is nice to be opening another Global IMMUNOTALK. If you're joining for the first time, I'm Elena Zúñiga from the University of California, San Diego, and I'm here with our fabulous Global Immuno speaker today, Dr. Akiko Iwasaki from Yale University. I would like to briefly remind you that next week's Global Immuno speaker is Dr. Kathy McCoy, so I hope that you can all join us. And also, I would like to highlight today that this talk is special because it's being featured as part of the program of the ongoing annual meeting of the International Cytokine and Interferon Society. So thank you so much to the Cytokine Society for featuring these Global IMMUNOTALKs in their program. Finally, I want to briefly mention that, as always, the questions for these Global IMMUNOTALKs are via Twitter. And so there is already in Twitter a tweet that is in the Global IMMUNOTALK account that says, ask questions for Dr. Iwasaki here. And you can reply to that tweet with your question and mentioning the hashtag Global Immuno. And Akiko has agreed to use her personal account to answer the questions. And so you can do this while you watch the talk live, or you can do this in an asynchronous manner if you watch the talk in YouTube. So without further ado, I would like then to introduce our terrific speaker today, Dr. Akiko Iwasaki. Akiko received her PhD from the University of Toronto in Canada in 1998, and her postdoctoral training from the National Institute of Health in USA from 1998 to 2000. She joined the Yale University as a faculty in 2000 and currently is an investigator of the Howard Hughes Medical Institute and Waldemar von Sedgwick's professor of the Department of Immunobiology and the Department of Molecular, Cellular, and Developmental Biology at Yale. As many of you know, Akiko is interested in how immunity is initiated and maintained at mucosal surfaces, which of course are important and natural ports of entry for pathogens. In particular, her lab focuses on understanding how viruses are recognized by the innate immune system and how that information is used to generate protective adaptive immunity. Akiko's studies encompass immune responses to several viruses, including herpes simplex virus in the genital tract and influenza virus, and more recently SARS-CoV-2 in the lung. With this focus, Akiko and her team have made major discoveries in the area of innate antiviral immunity and mucosal immunity that have resulted in paradigm shifts in our understanding of immune responses and vaccine design. It would be impossible to list all the impactful findings that Akiko and her group have made in this area without taking too much time of her talk that we are all looking forward to hear. So I want to keep this short, but also illustrate the high impact of Akiko's studies with few examples. Akiko was the first to demonstrate that DNA viruses are recognized by toll-like receptor 9 to produce type 1 interferons and cytokines. She was also the first to identify the critical role of autophagy in innate immune recognition of viruses and in antigen presentation. Akiko's and colleagues also demonstrated that the NLR inflammasome pathway is critical for host defense against influenza viruses. So these and other studies exemplify how Akiko's research really opened new ways of thinking about how cells recognize viruses and trigger the appropriate immune response to fight them. More recently, Akiko demonstrated that interferon induction depends on temperature. At the core body temperature, interferon induction is engaged maximally where the lower temperature found in the nasal cavity dampens this response. So this study provides an explanation for the long-standing and intriguing question as to why we catch a cold virus in cold weather. I would also like to highlight Akiko's studies that demonstrate the critical role for CD4 T cells in antiviral immunity as direct antiviral effectors, but also as gatekeepers favoring migration of CD8 T cells and antibody access to restricted tissues. And last but not least, and I promise I will not keep listing her findings, I would like to mention a breakthrough discovery that Akiko made showing that vaccines against local infections can be improved by establishing memory T cells at the exposure site. And this provided a new method called prime and pool, which relies on recruitment of T cells through chemokines and has a strong potential for clinical use to improve vaccination. So as you imagine, Akiko's work has been published in very impactful top-tier journals and in papers that are highly cited. She has also received many awards in recognitions for her tremendous advances in science. And particularly, I want to mention today the award she received in 2019 by the Cytokine and Interferon Society, named Seymour and Vivian Milstein Award for Excellence in Interferon and Cytokine Research. And also in 2011, she received the BD Bioscience Investigator Award from the American Association of Immunology. And again, these are just a couple of examples. So I'm looking forward to learn more about Akiko's research today during her talk that is entitled Immune Responses to SARS-CoV-2. So Akiko, I know that you're super busy and you may be one of the most demanded speakers. So we really appreciate that you have accepted our invitation to be a Global Immuno speaker today.
Thank you so much, Elena, for such a kind introduction, listing a lot of the things that we've done in the past. That's very special. Thank you so much. And I'm very honored to be one of the speakers in the Global IMMUNOTALK. I heard so much about this talk, and I know many people are watching and learning about the immune system. So thank you for your invitation, and Carla also for having me here.
We appreciate very much. So Akiko, as you know, we always ask a question to our speaker so we can learn more from them and also inspire young scientists. So the question we have prepared for you today is, what was the most challenging time throughout your scientific career and how did you cope with it?
The most challenging period of my scientific career is when I had my two children, and then when they were very young. I remember trying to balance family life and career and not having an on-site daycare access. We had to put our children in a daycare that was, a couple of towns away. So every time they would get sick or every time there's a problem, I had to drop everything and drive very fast on the highway to try to get to my children. And one of these times there were multiple phone calls that day. And on the highway, I just pulled over and started crying. I just thought I couldn't do this anymore. This was the closest I came to quitting science. And I also realized that there should be a better way of doing science. There should be a better infrastructure provided to parents of small children so that we can pursue science without this much trouble. And that's when I began my advocacy for women in science and underrepresented minority in science, because I realized how difficult it is to balance these things. And a lot of it can be solved with providing infrastructure. So that's was the very hard time, but I also learned my lessons from that, and I began doing something about it.
That's fantastic. Thank you so much, Akiko, for sharing that experience. Yeah, it is. I'm sure it's very inspiring for many females, including myself, to and all what you have accomplished. So thank you so much for all your efforts to pursue the career of women in science. I appreciate it.
Thank you.
Okay, so now if you can share your screen, we are looking forward to hear from your talk.
Thank you so much. I will share my screen. Okay. Oh, you can see this, yes?
Yes, perfect.
Okay, great. So I'm really delighted to be here today to speak to you about our recent work on SARS-CoV-2. So we began to work on the COVID-19 immunity as soon as the pandemic started in the US in March of 2020. And one of the learnings of this pandemic is that there is a huge variation in COVID-19 disease outcome. And of course, host age, sex, comorbidity status, pregnancy, all of these issues will affect the outcome. But even within a particular demographic group, there is variation. So this is one of the, fascinating aspect of COVID-19. And of course, it's also scary because being young and healthy doesn't guarantee that you will come out with a mild disease. So some of the things that we've done so far is to characterize immune responses in hospitalized patients who have moderate, severe, or lethal COVID, and really looking at the immune features that are associated with these types of outcomes, as well as looking at the neurologic symptoms, the amount of saliva viral load correlating with all these diseases, as well as autoimmunity. We've also focused on sex differences in immune responses to COVID, and particularly looking at the early phase of immune responses that distinguish some of the features between male and female patients, demonstrating, for instance, activation of T cells being much more robust in the female patients compared to male, whereas male patients appear to have a more inflammatory signature. We've also looked at the consequence of pregnancy in COVID and looking at some very severe case of COVID-19 infection in pregnancy, as well as even with mild infection having impact on the placenta and what that means, what that might mean in the future for coping with respiratory infection that might impact fetal health. And so these questions we had prior to COVID, we were engaged in different aspects of these questions, but COVID really provided us with a large sort of platform and patient base to look into these mechanisms. So today what I'd like to do is to talk about 4 different stories. The first one being about timing of neutralizing antibodies. Second one is to look at how different variants that are circulating might impact the immune protection conferred by vaccines. A third story is about a mouse model, using mouse model to dissect the different effector arms of the adaptive immune response in fighting SARS-CoV-2 infection. And finally, I'd like to talk about current and future plans for looking at long COVID. The first study was led by Carolina Lucas and John Klein on the timing of neutralizing antibody production. All of our human and translational immune studies are enabled by the biorepository called Impact Yale, which was established by Professor Albert Ko as the lead, where we were able to collect biospecimen from patients that are in the hospital, cohort 1, patients who are outpatients, cohort 2, as well as healthcare workers in a longitudinal manner. And this enabled us to study immune response in real time and in the outcome of disease in these patients. And so we collected blood, saliva, nasopharyngeal, oropharyngeal swabs, as well as feces and urine from the inpatient cohort every 3 to 4 days. And similarly, from outpatients, we were able to collect biospecimens And the healthcare workers also, we provided daily testing from their nasopharynx and saliva to monitor for any potential exposure so they can isolate themselves in the case of positive results. So the study that I'm about to tell you really began by looking at what is really correlating with lethality from COVID with respect to the immune response. And here on the left panel, demonstrating the anti-spike IgM or IgG. On the right panel is the anti-receptor binding domain of spike, IgM and IgG. And the black dots represent negative control, the healthcare workers that are not infected. And if you look at their value, of course, there's no reactivity to the SARS-CoV-2 spike. Whereas the non-hospitalized, the dark purple dots indicate patients with milder disease. And these patients do develop, for the most part, IgG against the spike, but not as much as the moderate patients in the hospital or the severe patients who require ICU in the hospital. So there's a disease severity dependent induction of IgG against the spike in RBD, which is also reported by others. And we also see that the disease patients, it's not that they fail to develop antibodies, but they still succumb to this infection. So what appears to distinguish the disease patient from discharged patients is the amount of viral RNA we find in these patients over time. So the lethal cases indicated in purple here, they maintain high levels of NP viral load, even out to 30 days post-symptom onset, whereas those patients who discharged alive are able to gradually control the viral load. And when you look at the viral load at maximum antibody titer on the right from the discharged patients, that viral load is much lower than those patients who have lethal COVID in purple. We also had a few patients we call high neutralizers who had high levels of neutralizing antibodies from the onset of the study. And these patients had very low levels of viral load. And what's striking about the difference between the deceased and discharged patient is the time course with which they develop neutralizing antibodies. So if you look on the right panel, this is the IgG against the spike on the top and RBD on the bottom. You see that the deceased patients, they do develop a maximum level of IgG against these antigens, but it takes them a long time to get there. Within about 30 to 35 days, they reach this maximum level, but they really do fail to do so early on during the disease. Whereas people who have been discharged from the hospital alive, they're able to mount antibody responses within about a couple of weeks to reach maximum titer. And as I said, there are a few people who had high neutralizing antibody from the beginning of the study, and they had high levels of antibody against the spike to begin with. And they really have maximal anti-RBD level from the very beginning. And these patients are able to clear the virus quite well. So then Carolina decided to divide patients into two groups, one that had early neutralizing antibodies who developed neutralizing titers prior to the 14 days of disease onset, whereas late neutralizers did so after the 14 days. And she followed the clinical outcome of these patients over time on the right. And you can see that about after fourth collection time point, patients who had early neutralizing antibodies tend to control disease and are become discharged. So 0 clinical score means discharge and 6 means death. Whereas the late neutralizers, they're very similar in the first 3 time collection points, but around the 4th collection point, these patients unfortunately develop worse and worse disease outcome. And what's different about these patients? Well, there is very little difference in the age, spread of the age group in these patients. So the early neutralizer in pink versus the late neutralizer, they're about similar age groups, as well as BMI. And there's no difference in the gender frequency between the early versus late neutralizers. However, there is an increase in the mortality frequency in the late neutralizers having more of this, as well as NP viral load being higher in the late neutralizers. So what this study demonstrated is that the COVID patients with lethal disease had delayed antibody onset compared to those who were discharged alive. And the maximum antibody levels were not very informative in distinguishing patient disease outcome. And patients who developed neutralizing antibodies 14 days or earlier were able to, in general, control disease, while those who developed it later than 14 days were more enriched in the lethal group. And so this study does raise some interesting questions about the role of antibodies in different phases of COVID-19 disease. And in particular, it also has an implication about the use of convalescent plasma or monoclonal antibody therapy, that our studies support earlier use of these therapies to benefit the patient, because later on, what we see in the natural course of event is that these types of antibodies may not be useful. And that's essentially what the clinical trials are finding so far, that the early use of monoclonal antibody cocktail, for example, has much more benefit than late use. So we've been learning a lot about this immunity during the course of infection, but we also wanted to turn our attention to vaccine-induced immunity. In specific, we wanted to address how variants of SARS-CoV-2 affect vaccine-induced immunity. And this study, again, was led by Carolina Lucas, along with two other first authors, Dr. Chantal Vogels, who is a postdoctoral fellow in Nathan Grubal's lab, who is doing SARS-CoV-2 surveillance, and Dr. Incy Eldrum, who is a pediatric infectious disease doctor who contributed equally to this study. So the design of the study is to recruit healthcare workers who were either infected previously and recovered, these are the blue people, and people who were never exposed to SARS-CoV-2, the purple people. And we collected their blood prior to vaccination, as well as 7 days and 28 days post first dose, and 7, 28 days or 70 days after second dose of mRNA vaccines. So the good news is that the mRNA vaccines elicited very robust antibody response to the ancestral spike protein. So here, the top row represents anti-spike IgG, the middle one anti-S1 region, and anti-RBD on the bottom. But regardless of what you look at, you see that after the second shot, everyone generates very robust antibody against the spike. And this, of course, depended earlier in the previous exposure status. So people who had previous exposure to the virus, which is indicated by blue dots here, they started off with some levels of antibody against the spike. But then even after the first shot, these people were able to elevate quite significantly their level of anti-spike, and this was maintained at high levels throughout. However, even in those previously uninfected people, 2 shots of vaccine ultimately generated similar levels of antibody against the ancestral spike protein. And also a good news is that the mRNA vaccines, after 2 doses, were able to generate robust neutralizing antibodies using authentic SARS-CoV-2, the ancestral strain. And here on the left is an aggregate of everyone that we monitored. And you can see that vast majority of people are able to elicit good neutralizing titers against that SARS-CoV-2. And on the right, we divided again into people who had prior exposure in blue, versus who's never encountered the virus in purple. But again, after the second dose, everyone except for 2 people are able to generate a very high neutralizing antibody against the SARS-CoV-2. We also looked at T cell immunity that's generated by the vaccine. On the top of this graph are the CD4 T cells, on the bottom, the CD8 T cells. And here we tested T cell reactivity against 2 different lineages of spike, the original one in lineage A, as well as the P1 variant of concern, which is the gamma variant that originated in Brazil. And we were able to see that the CD4 T cells, regardless of whether people were previously infected or not, after 2 doses of mRNA vaccine, there's a robust CD4 T cell response and CD8 T cell response. The only difference we saw was that for the CD8 T cells, the P1 spike variant elicited a slightly less activated T cell phenotype compared to, say, the original strain. But for the most part, T cell responses are preserved against the variants in both CD4 and CD8 compartments. We then looked at the ability of vaccine-induced antibodies to neutralize different variants of interest and variants of concern. So Carolina and Chantal did a heroic effort of isolating, purifying, and amplifying different variants that are circulating in the community, as well as acquiring some from other places to be able to study neutralizing antibody activity against authentic viruses. So here on the left is the different mutations that are found within the spike domain of these different variants, and variants are listed in different rows. And on the right of this panel is the neutralizing titer of antibodies generated by the vaccines. So what you see is, again, as I just told you, against the original strain, many people, most people except for 2, are able to generate very good neutralizing titer. However, if you go down the list, you start to see decline in neutralizing capacity starting around Delta. So the Delta variant harbor several mutations, one of which is the L452R mutation. And this particular mutation has the most impact on escape from neutralization, as shown on the right. But however, you go down the list and you start to see that other variants have even more capacity to escape the neutralization, such as the gamma and the beta variants being the most to escape the neutralization. And these viruses, in addition, have multiple mutations, such as the famous E484K, as well as N501Y mutations, as well as many others that are in combination increasing the escape. But what was striking to us is that when we looked at the inter-individual variations in neutralizing antibody responses, there was such a huge spread in terms of whether a person generates high levels of neutralization after vaccination. So here is, again, the different variants that are listed on the bottom and neutralization titer of a given individual. So in general, if a person is able to generate very high levels in the thousands of the neutralization titer, they tend to remain high levels of such neutralization across the different variants. However, people who started out with low levels of neutralization drop off with respect to their ability to neutralize some of these more highly escaped variants. And as I mentioned, a couple of people just failed to generate any neutralizing titer against any of these viruses. And once again, when you divide these participants into previously uninfected on the top and previously infected on bottom, what you see is that the drop-off in terms of the ability to neutralize different variants happen more quickly for the previously unexposed versus those who are previously infected. And this is really no surprise to any immunologist because the previous infection confers just another exposure to the antigen and, really serving as the first dose out of a 3 exposure, whereas the non-previously infected, these people only had the mRNA vaccine twice. So hopefully the third dose is able to bring everybody up to this level down here. So this study demonstrated that the mRNA vaccines do elicit robust antibody, neutralizing antibodies and T cell responses to the spike protein. And prior infection gives people advantage to generate even higher neutralizing antibodies to both ancestral and variants of concern. And we also found that distinct mutations confer different degree of escape from neutralizing antibody. And there are also certain combinations that synergize or antagonize this escape. And finally, we think that the viral genome surveillance effort globally are critical to understand the transmission and breakthrough potential of variants of concern. And having this sort of identified different mutations having escape capacity, we may be able to predict whether a particular variant will be more able to escape neutralizing antibodies from vaccines or potentially prior infection. So I talked a lot about the neutralizing antibodies, but of course there are other effector function of adaptive immunity that can confer protection. And so Ben Israelo, who is an infectious disease fellow in the lab, decided to tackle this question. What are the determinants of protective immunity, adaptive protective immunity against SARS-CoV-2? And he made use of an animal model that he created before to study this question. And so, as I mentioned earlier, there's a lot of studies, including our own, that correlate the neutralization antibody levels to protections from infection or disease. And this is a very nice study that demonstrated different vaccines or prior infection with respect to how much neutralizing antibodies they generate, and which correlates with protective efficacy of each of these vaccines. And there's also non-neutralizing levels of antibodies that protect from SARS-CoV-2 infection. A study by Gali Alter, for example, looks at the FC region of the antibodies generated by the vaccines and how they confer protective immunity. And of course, T cells are absolutely critical, not only in inducing the antibody response, but also serving as effectors to provide protective immunity. So using a mouse model, we can actually dissect this question much more readily. And the first question Ben asked was, what is the role of B and T cells in clearance of from the primary SARS-CoV-2 infection? So previous to this, Ben developed a mouse model in which he can transduce any animals of any genetic background with AAV encoding human ACE2, which is the receptor for SARS-CoV-2. And after about 14 days, these animals become susceptible to infection with SARS-CoV-2, any human isolates that we have in hand. So what he did is he transduced animals, either wild type or the MUMT mice, which are devoid of mature B cells, and challenged these animals after transducing with human ACE2 with SARS-CoV-2 and monitored viral load over time in the lung. On the bottom is the infectious viral particle per mil within the lung. So the wild type animals are able to completely control infectious virus within 7 days. The MUMT mice was strikingly capable of controlling the primary infection. Within 14 days, there are no infectious particles. Within 7 days, there's a little bit of infectious particles left, indicating that B cells are not necessary to control primary infection. However, when Ben did the same experiment in the rag knockout mice, in which there are no T or B cells, these animals were unable to clear the primary infection and they became chronic carrier of the virus. So this indicated that you do need adaptive immunity to control primary infection. Next, Ben wanted to understand the role of T cell subsets in clearance of the primary infection. To this end, he transduced again the animals with ACE2, and then 2 weeks later, challenged these animals with SARS-CoV-2, either in the presence or absence of anti-CD8 or anti-CD4 antibodies to deplete the CD8s and CD4 subsets. So on the top right here, this is the infectious viral particle obtained from the lung at 14 days post-challenge in wild-type animals that have been either PBS treated, CD8 depleted, CD4 depleted, or depleted of both. And what you see is that the depletion of CD8 alone or CD4 alone does not impact the ability of the animal to control primary infection. However, depletion of both CD8s and CD4s did lead to some detectable virus in some of the animals, indicating that T cells are required for the complete clearance of the virus. Now, when Ben looked at the role of these T cells in antibody response, the depletion of the CD4s and CD8 led to the most dramatic reduction in the antibody. So this may also relate to the antibody responses that are at play as well. And to really further address that possibility, Ben did the same experiment in the MUMT animals. So as I mentioned earlier, MUMT animals are able to clear the virus completely on their own within 14 days. However, when you deplete CD8s from these animals, you start to see some animals still having infectious virus in the lung at 14 days, indicating that if you just have the CD4 T cells, they're not sufficient to completely clear the virus. And similarly, CD4 depletion in the MUMT mice led to some of the animals, again, having some virus, meaning that the CD8s alone are not sufficient to clear the primary infection. And of course, when he depleted both CD4s and CD8s from the MUMT, this sort of phenocopied the rag knockout mice that he observed, and these animals are unable to control the virus. So then he turned to the question of the role of B and T cells in protection against reinfection or vaccine-mediated protection. The first approach he took to address this question was to do an adoptive transfer of either immune sera or T cells into a AAV transduced rag knockout mice. So in these recipients, essentially all you have is the adoptively transferred sera or adoptive transferred T cells to fight off the infection with respect to adaptive immunity. And he then infected these animals with SARS-CoV-2 and measured viral load in the lung at 7 days. So this panel C demonstrates that if you didn't have any transfer, of course, these rag knockouts cannot clear the primary infection. However, T cell transfer led to reduction in the viral load in the lung, as well as some of the animals being able to completely clear the infection, indicating that T cells are partly sufficient for clearance of the virus. However, when he transferred this immune sera, all of these animals completely controlled infectious particles in the lung, indicating that the antibodies are conferring protective immunity, sufficient for conferring protective immunity against SARS-CoV-2. Ben then turned to a interesting model in which he created a situation of animals having different levels of adaptive immunity against SARS-CoV-2 by immunizing mice, this time K18 mice. These are the transgenic animals that express human ACE2 under the K18 promoter. And he immunized these animals with 1 microgram all the way down to 0.01 microgram of mRNA vaccine. What this did was it allowed him to generate a condition in which animals had different levels of anti-spike IgG. So there's a beautiful dose response with respect to having 1 microgram, 0.3, 0.1, 0.03, and 0.01 microgram of mRNA vaccine generating distinct levels of antibodies. When he challenged these animals with SARS-CoV-2, he saw that animals that received 1 or 0.3 microgram of mRNA vaccines were completely protected from disease or death, whereas those that received 0.1 microgram in the green, they were partly protected from disease and death. And the ones that received 0.03 or 0.01 were essentially the same as the mock immunized animals. So this allowed Ben to look at the correlative protection with respect to death or disease by sort of correlating the level of antibodies to those outcomes. And he was able to do this. On the right, what I'm showing you is the level of IgG on the x-axis here against the spike 1 domain, and the percent starting weight on the y-axis. And there's a beautiful correlation between the amount of antibody an animal generates to the sort of prevention of weight loss that they have against the disease. And similarly, he calculated the levels for neutralizing antibodies on the bottom. So there is a very nice correlate of protection based on the amount of antibodies that the animals were able to generate. Finally, Ben asked the question of the requirement of CD8 T cells in conferring protection against either reinfection or vaccine-mediated a sort of breakthrough infection. And to do this, he again took the K18 animals and then created an mRNA vaccine cohort, which received 2 doses of mRNA vaccines. And then also the convalescent cohort, which received sublethal dose of SARS-CoV-2. And these animals, after a certain number of days, were then challenged with SARS-CoV-2, either the original strain or the beta variant. And prior to challenge, he either depleted CD8s or not from the host. So the vaccine and prior infection induced very good levels of anti-spike IgG, shown here, as well as neutralizing antibodies, even though the vaccines provided higher levels of these antibodies. When these animals were challenged, all of the mice that were either prior infected or immunized with mRNA vaccines were completely protected from disease or death, whereas all the naive animals challenged at the same time died within 4 days because we used a high-dose challenge model here. And the good news is that within 2 days of challenge, when Ben looked for infectious viral particle, vaccinated mice as well as convalescent mice completely cleared any infectious particle from the lung, whether they were depleted of CD8 T cells or not, and whether they were challenged with original strain or the beta variant. So this study demonstrated that T and B cells both contribute to the clearance of the primary infection, and that in the absence of one, the other can compensate. However, if you're lacking both T and B cells, animals are unable to control primary infection and remain chronically infected. And the adaptive transfer study demonstrated the complete capacity of the serum and partial capacity of the T cells to clear the primary infection. And in both the setting of convalescence as well as mRNA vaccine, CD8 T cells were not required for protection. And finally, antibody levels and neutralizing titers correlated with immune-mediated protection. So this study illustrates this very sort of fantastic feature of the mRNA vaccines in inducing not only systemic immunity, but some crossover to mucosal immunity, For instance, there are studies now demonstrating that you can find neutralizing antibodies of both IgG and IgA isotypes in the mucosa of the vaccinated people. And so this will prevent infection altogether. And even if infection was to take place, there are lots of other effector molecules and cell types that can control the spread of the virus. And even if that were to happen, the vaccine-induced immunity will prevent a lot of the transmission-capable activities, such as by preventing disease that lead to sneezing and coughing, as well as potentially the emitted virus from a vaccinated person might be less capable of infecting the next person if they're coated with antibodies. So there's a lot of different paths to controlling infection and disease provided by the mRNA vaccines. And they weren't intended to generate mucosal immunity, but they ended up doing so. And future or next generation vaccines might benefit by specifically triggering the mucosal immunity, as Elena mentioned, by using something like prime and pull. So finally, I want to spend the last few minutes talking about long-term consequences of COVID. We believe there are at least two types of long COVID that can occur. One is from patients who were discharged from the hospital who had either moderate to severe COVID. And these patients are now known to have at least half of them having prolonged symptoms over months after discharge. And these long-term consequences may be triggered by the fibrosis, tissue damage, organ damage, or even post-ICU syndrome, which is well known to occur regardless of the cause of ICU admission. Thus, the second type of long COVID is seen in patients who never were hospitalized, but they had milder disease. And these patients no longer have detectable virus, actually top and bottom have no longer detectable virus in the nose or the saliva. However, about 10 to 30% of these patients do develop long-term symptoms. And we can think about these patients as having post-COVID-19 fatigue syndrome, in which they have chronic and severe fatigue, brain fog, post-exertional malaise, as well as dysautonomia. Both types of long COVID is very important with respect to studying and understanding the pathogenesis, but they may have different types of pathogenesis depending on which of these types they belong to. And the one that I'm going to focus on today is the latter part, these post-COVID fatigue syndrome. If you look at the different papers reporting this kind of long COVID, there are over 200 symptoms associated with long COVID, including systemic impact, and many of them have neuropsychiatric impact, the head, eye, ear, nose, and throat, endocrine, cardiovascular, pulmonary, musculoskeletal, and GI tract, and many others. And so how can a respiratory infection result in all of these involvement of all these organs? Well, we don't know the answer to this. Here is just sort of a snapshot of looking at like how many people maintain these kinds of long-term symptoms. And here on the left is showing that the severe to very severe symptoms do wane over time, but this is replaced by mild to moderate disease symptoms that persist for well over 6 months. On the right, I'm showing you that patients who started with average of, let's say, 11 symptoms, they tend to decline over time and recover within 90 days, whereas those patients who started with more than 13 symptoms continue to accumulate different symptoms, and they are still maintaining a large number of symptoms over 6 months. So how does long COVID happen? Well, there are two main hypotheses that we are currently pursuing. One is a establishment of viral reservoir in different tissues, And this can be either infectious virus or viral remnants, RNA, protein, or mixture thereof that can stay in different tissues and trigger chronic inflammation. And on the right is the possibility of autoantibodies and autoreactive T cells that target different tissues. And there's also tissue damage, dysbiosis, and latent viral reactivation. These are all possibilities that contribute to long COVID. So with respect to the two main hypotheses, the viral reservoir and autoimmunity, there's ample evidence coming out that both of these things can be happening. The first example is a study that demonstrated that in the intestinal biopsies of patients who had COVID, months ago, still maintain viral antigen in the enterocytes here. You can see this is a nucleocapsid staining that you can see within the intestinal biopsies. And so 5 out of 14 patients had positive staining. 3 out of 14 had PCR positive for viral RNA after 3 months. Another report that looked at surgical specimen from patients who had COVID 9 to 180 days before were tested positive with respect to either viral protein or viral RNA. They found it in various tissues, including colon, appendix, ileum, hemorrhoid, liver, gallbladder, and lymph nodes. So this provides this picture that even though it's a respiratory infection, the virus RNA, virus protein can be found on multiple different tissues. And we and others have also reported autoantibodies that developed during acute COVID disease. And this study I highlight here is collaboration with Professor Aaron Ring's lab at Yale, where he and his team and my lab collaborated together to look for autoantibodies in acute COVID. And this was enabled by the technology that Aaron's lab developed, which is called Rapid Extracellular Antigen Profiling, or REAP, where he developed yeast display library expressing thousands of human exoproteome, which are secreted or transmembrane proteins displayed on the surface of the yeast. And this can be combined with IgG from the patient. And then you can pull down IgG-bound cells using magnetic beads, and you can sort of decode which antigen that these cells produced as autoantigens using deep sequencing. And using this REAP technology, we identify numerous autoantigens that either target immune cells, immune factors, or tissue-associated antigens that are indicated here that are expressed in the brain, the vasculature, connective tissue, and many others. So each of the columns represent patients, and each row is an autoantigen. And you can see that the brighter the yellow color, the more autoantibody there is in that particular patient. And there's a large number of these autoantigens that are picked up by antibodies from these patients. And there are many other reports demonstrating autoantibodies in both acute and long COVID now. So this demonstrates that autoimmunity may be triggered by the SARS-CoV-2 infection. So currently there are no approved or effective treatment against long COVID. However, on Twitter, I started seeing lots of patients reporting symptom improvement after getting the vaccine. So this is a very nice report from a patient-led group called Long COVID SOS that surveyed the patients for symptoms after the vaccination in long haulers. And what they reported is that about 57% of the patients had improvement in their symptom, while 19% had worsening. And breaking down the different symptoms, you can see that each vaccine has different impact with respect to improvement for fever and muscle pain and so on and so forth. But this study really illustrated that vaccines may have an impact on long COVID. So how can vaccines have an impact on long COVID? Well, going back to the hypothesis that either viral reservoir or autoimmunity is leading to a long COVID, you can imagine that the vaccine elicits robust antibodies and T cells that can control the reservoir or even eliminate it. And that would provide a permanent solution. Whereas it's also possible that vaccine-induced cytokines and interferons, either coming from innate recognition of the vaccine particles or adaptive immune response to the viral antigen, you can imagine these kinds of cytokines having some regulatory impact on the T and B cells to temporarily shut down these kinds of pathologies. So currently, we are collaborating with a large number of groups to try to understand immunological and physiological determinants of disease pathogenesis in long COVID. And the goal is to inform rational diagnosis and therapy. So the way in which we're doing this is it's a large collaboration with multiple different patient-led groups, as well as the physicians that are treating these patients. And we're collecting blood and saliva from these patients, looking in real time for different activation markers and cellular subset from PBMC, as well as doing further deep profiling of cytokines and autoantibodies, T cell receptor usage, metabolomics, and hormones from these patients, to finally be able to integrate these types of multimodal data using computational approaches and machine learning. And our ultimate goal is to share these data back to the patients. And obviously, the ultimate goal is to find a better diagnosis and therapy for long COVID, which millions of people are currently suffering from. So I want to end here by thanking all the authors that contributed to the studies that I was able to share with you today, as well as my entire lab members who've been working selflessly to look for not only just the immune response to SARS-CoV-2, but participating in early days for surveillance of the healthcare workers and impact team, without whom none of the translational work could have been done, and many other teams that we're collaborating to understand pathogenesis of long COVID. And of course, this is my lab celebrating full vaccination status in the summertime. So thank you so much for your attention.
Thank you so much, Akiko. This was wonderful talk, and thank you so much for all your important research and all the efforts you've done to advance our understanding of disease. Yes, thank you very, very much. And I'd like to share again my screen so I can remind people that questions in Global IMMUNOTALKS are via Twitter, and that if you search for the account Global IMMUNOTALKS in Twitter, you will find a tweet that says ask questions for Dr. Akiko Iwasaki here. You can reply to that tweet, and Akiko will use her own personal Twitter account, Virus is Immunity, to answer those questions. And also you can ask questions in an asynchronous manner after watching the talk in YouTube, if that was your choice. Thank you again, Akiko. Really appreciate your efforts, and it's an honor to have had you as a Global Immuno speaker today. Thank you very much.
Thank you very much. Thank you, everyone.
Bye, everyone. See you next week.